or do you have any questions?
I do have a question. What kind of primitive primates do you use for your work?
We don't use any primates at all.
Or I mean, primitive animals.
Primitive animals. I use lamprey, which are basal vertebrates, and so this is actually that's an interesting story because I didn't know very much about other vertebrates. We were really concentrating on using chickens and mice and frogs mostly for the experiments that we did. And I was teaching in a course at Woods Hole where they have good graduate courses. You guys should go there, after you finish, because they have some excellent courses.
Woods Hole.
Yeah, and the embryology course there starts with looking at very. Sounds like maybe it's this guy. We started with very primitive animals, like sea urchins and other marine life, and you sort of work all your way up to mice. And I got really interested from that in trying to understand where neural crest cells came from in an evolutionary sense. So we decided to look for animals that were sort of at the base of the vertebrate tree, and that's when we discovered jawless vertebrates like lamprey and hagfish. And these, I don't know if you've ever seen a lamprey. They're really ugly.
Right.
They have, I might be able to find a picture for you. They have
We'll get one online here.
Sort of a modified jaw, and they are parasites, so they eat fish, and they just sort of glom onto the fish and they eat them. Let's see. Are you Googling something?
We're trying to pull it up online, but at one point they were predators, is that correct?
They are, yeah,
They were predators, now they are scavengers.
Yeah, well, no, the hagfish are scavengers, but lamprey are actually parasites.
Oh, okay.
I have a picture right here.
Ooh, we've got one right here. They are ugly. They're like something out of a horror movie. Look, there's one guy on the guy's head.
Yeah, so we decided to develop these in the lab. Let's see if I can find my
Charming. Do they saute well with garlic?
Yeah, that's the first. I got a recipe, so I don't know.
Oh yes. Charming.
That's them in the lab. Anyway, so we've been working with those, and they only breed once a year for about 2 weeks. So one of the real challenges was getting them to be a good experimental model. So we found out that by controlling their temperature, we can actually expand their breeding time. And so now we can, for about 2 months of the year, we have these animals and they produce embryos in the lab. And so we can use them almost like people do frogs. And we have scientists coming from all over the world to work on them because this is one of the few places where you can get embryos.
You've got them. And so your lab is full of aquariums and
yeah, well, we have this one room where we have 3 different tanks. These things are big. So, we get them by FedEx, and during the summer it's easy to get them because people want to get rid of them. So.
Oh boy, yeah, they don't look like very friendly pets.
They're not. So that's what we're using. So we use lots of different animals though. So I have zebrafish in the lab, xenopus, which is a type of clawed frog, chickens, and sometimes we use mice though. I try not to very often because they're expensive, and also they're cute and furries.
Right. Getting attached to your animals.
So what are you guys interested in? I'm very, first of all, I'm very impressed that you formed this club. I think that's really cool. And I was really interested in how this came to be and, why you decided to interview women scientists.
Well, it's kind of a collective, and we're a support group together, and there are 2 other neuroscience clubs on campus, and they just sit around and read papers. And we had access to this fabulous high-tech room, and we're just building the club. We're trying to put on a dance in a month or so, and so we're trying to be a club and have fun and do some serious investigative work. But and we look at women researchers because we find there's a big difference in how they approach issues and research and their methods and such, and it's real refreshing. I find it, and then I can I personally go into other research by non-women, and it's kind of striking the difference between how women look at research So it's good. It's all good. And we're not exclusive, but we're just interested in how women think and how.
No, I find it very different. I mean, I know the way I run my lab is I sort of run it like I would my family, and you know, I try to be really nurturing, and I find that it works. You know, you're nice if you're nice to people, they actually work harder and they try to please you more. It's sort of interesting. So I have to call up people in my lab and say, please go home. It's way too late. You shouldn't be working this late.
But we love you, they say.
So it's funny.
Well, you were talking about the how it has to relate to the sensory system and things like that. And I'm interested in the ear personally, so I was wondering if you had looked further into the senses or.
Yeah, well, I have to say I'm not really interested in the senses. I sort of like to know how the structures develop. So I pretty much focus my interest on the early stages of development. But we know quite a bit about the ear, and it's actually, you were asking about relationship between placode and neural crest. And so the ear is the otic placode. And it turns out that a lot of the same genes are expressed in both tissues, and we're trying to understand how why the same gene does different things in different tissues, and it's really fascinating because in some cases we found that you've got the same thing functioning, but the kind of genes that turn it on are different, the inputs are different, and so there are these fascinating relationships between the two tissues. The otic placode not only forms the ear, but it also forms acoustic ganglia, and that also gets a neural crest component. The glia come from neural crest. So there are all these interactions between the two cell types. And then the neural crest cells form the bones in the ear also. So the two things seem always work together and they're coordinated. So there's some very interesting labs working on later stages in ear development. If you're interested in that, I know two women actually who are working on ear, both of them are very good. One's name is Doris Wu, and she's at the NIH. And the other is Donna Fekete, spelled Fekete. I think Donna just moved. I think she was at Purdue, but I'm not sure where she is now. I think
we'll look them up.
Well, yeah, they're both really nice people.
Before I came over into biopsychology, I was a professional singer and musician, and my interest in neural crest is that it seems to be land-based phonation. And so I noticed there was, oh gosh, I've taken so many notes on this, I don't know if I can keep it all together, but the only thing that throws me off are the pigment cells, but the whole sensory aspect of sound and resonance on land seem to center around these derivatives, including the auditory and even vestibular, standing upright and being functional in a land-based situation.
Yeah, but neural crest cells came way before we got onto the land, right? Oh. So, I mean, fish have perfectly good neural crest. They have all the derivatives we have. Right. So the thing about the neural crest is they keep changing with time. It's this whole evolutionary issue. They seem to evolve more quickly than other things. And so I think that it probably helps facilitate some of these land-based skills, but there certainly are very good precursors to them in all these other organisms. If you look at reptilian ears, they have pretty good ears, but the bones have been modified in mammals.
Right.
So,
it's an interesting evolution to follow those auditory bones. And what are the transcription factors that enable this morphology in those cells? Is that snail 1 and snail 2 is just the beginning, I'm sure.
Yeah. Well, so most of what we've looked at are transcription factors that function fairly early, and those are very ancient. They're conserved in lamprey and in us. So the changes that happen probably happen quite late and at sort of advanced stages of differentiation. I mean, we really struggled to find things that were different between lamprey and us, which is sort of embarrassing because when you look at these animals, they're really ugly. You don't want to look like them. But because we're looking at the very early aspects, it it means that those things are very old, and the modifications that have occurred have probably occurred on top of them. So the things we're looking at, like snail, are going to be everywhere, in every organism. But you can imagine if you want to modify the bones of the ear, for example, what you're going to be doing is something that's quite late, and when those bones are beginning to condense and then ossify. So there are many layers of sort of gene regulation that occur before you get to that point. And we don't know very much yet about the changes that are occurring, though people are studying those. And that's where I think doing this comparative type of approach, where you look at many different living organisms, can give you an idea of how these changes might have occurred. Yeah, I've gotten fascinated by that recently. It's hard, though. The thing about studying how things have changed over evolutionary time is that you can never go back and look at that ancestor and see what the ancestor looked like. So there's two things. You can never be wrong, which is good, but you can never be right either.
No, not for sure, can you? So these crest cells respond to local environment cues, and so the macro level, we're just talking about that with phonation and sensory needs. Does neural crest, do the derivatives have a connection to genetic expression? As they transform so quickly and readily to the environment, my question is, what do you see as that having a connection to our conscious awareness? Do you think that
I have no idea.
consciousness is such a big thing and transformation? So
that's a really difficult question. And one of the reasons I like studying what I do is because at the time that the embryos that I'm studying are really simple compared to us. If you look at our brains, they're so complex that it would be wonderful to understand how. I'd love to understand the development of the brain of a newborn, but we have no understanding of that because even that is so complex. And if you see how when a baby is born, they can't do anything. So the connections have not yet formed. There's some kind of consciousness, but it's mostly about getting fed, and so really. I don't know very much about consciousness. Some of the people in my department actually work on that, and I see it as being at least 100 years away from having in-depth understanding and knowledge of. So, and in terms of neural crest, the good thing about neural crest is it really represents our, sort of things that are structural. The most, I guess one of the things that neural crest is responsible for is like fight or flight behavior, and that's probably the most conscious thing or unconscious thing I can think of. It doesn't really relate to, thinking about what you want to do.